Oh, damn. Hi, good morning. We're going to start in just a minute. Good afternoon. I'm Jennifer Gross, CEO of American Friends of Tel Aviv University. I hope you and your families continue to be healthy and well during this ongoing time of social distancing and working remotely. I'm pleased to welcome you to today's webinar, From Cancer Immunotherapy to COVID-19 Vaccine, featuring Professor Ronit Satchi-Fainaro. We are grateful to Professor Satchi-Fainaro for taking time out of the lab to speak with us today about how she and her team are converting the nano vaccine that she developed for melanoma to a vaccine for COVID-19. Professor Satchi-Fainaro is head of the Cancer Research and Nanomedicine Laboratory and head of the Tel Aviv University Khan 3D Printing Initiative. She holds the Kurt and Herman Lyon Chair in Nanosciences and Nanotechnologies. Professor Satchi-Fainaro received her degree in pharma from the Hebrew University, her PhD in polymer chemistry and cancer nanomedicine from the University of London. She then spent 4 years as a postdoctoral fellow, a research fellow at Harvard University and Children's Hospital in Boston, and joined Tel Aviv University in 2006. Professor Satchi-Fainaro's multidisciplinary lab focuses on basic research, elucidating the mechanisms underlying the switch from cancer dormancy, leading to the discovery of new molecular targets to interrupt tumor-host interactions. She has published more than 110 manuscripts, 12 book chapters, edited 2 books, and is named inventor on 55 patents, some of which were licensed to pharmaceutical and biotech companies. Her scientific achievements have been acknowledged numerous times by leading magazines such as Forbes, and Professor Satchi-Fainaro was named Globe's Woman of the Year in 2019 and was named on Israel's Top 40 Under 40 by The Marker and other publications. Following our conversation, Professor Satchi-Fainaro will take questions which you can ask in the Q&A box on your screen. Professor Satchi-Fainaro.
Thank you, Jennifer, for the kind words, and thank you for inviting me to share a little bit of what we do in normal times before the pandemic, the COVID-19 came to us. But I will also share some of the insights that we collected in the last 4 months, taking everything that we know and developed for cancer and cancer immunotherapy and trying to exploit that for creating a new COVID-19 vaccine. So back to cancer, I will tell you where we started when I opened my lab in 2006 at Tel Aviv University at the Faculty of Medicine. I just came back from a postdoctoral fellowship in Boston at Harvard Medical School, working with Judah Folkman there on tumor dormancy, amongst other things that we did then. And what really turned on my curiosity was a study published in 1993, a Swedish study on 16,000 people that died of trauma. Those are people that died of car accident, suicide, anything that is traumatic, nothing to do with any disease that they were aware of, definitely not cancer. And I found the results striking. They found that about 46% of the men had these tiny lesions in their prostate. I'm talking about a 1 to 3 millimeter in size, in situ carcinomas. In situ, this is in Latin for in sight, very local small lesions. Then about 39% of the women at this age range had these tiny lesions in their breast, and about all of them, 99.9%, had these tiny in situ carcinomas in their thyroid. But I'm not here to depress you and think that all of us have cancer, but rather thinking these are dormant tumors that were never awakened. Meaning that if we look at how many in these populations, which percent are actually tumors that were diagnosed clinically, meaning that they had symptoms. For a symptomatic disease, we had only 1% of men in the ages of 60 to 70 present with prostate cancer, 1% of women with breast cancer, and only 0.1% of thyroid cancers developed. So in a way, maybe we should think a bit differently about cancer. Instead of thinking why people get cancer, and this is present company included, I was working in the same way with the same research approach for years, maybe we should think why not? Because maybe it means that even though we have the potential from those dormant tumors to have a progression for a symptomatic disease, in most cases, as I showed you with these low percentages, in most cases they remain dormant and we live until the age of 120 without even knowing that we have these dormant lesions of cancer. So if we find out what in our body keeps those these lesions dormant, maybe we can prevent cancer, prevent their awakening and their progression to metastatic disease. Because most times we know how to deal with primary tumor, but not with the systemic disease, meaning that it already spread as metastasis. So if we look at the first stages of tumor development, there are the two-hit theory, meaning that there are two main kind of mutations that transform a normal cell to a cancer cell. The first one is the one that tells the normal cell now to start dividing without control, becoming immortal. And the second is to stop dying, meaning that we have a QC system, a quality control or quality assurance, like a regulator in each cell that tells the cell to die if it finds that something is wrong. In a way, it's called apoptosis, coming from Greek, for foliage. This is what happens to the trees when the leaves fall, but keeping the tree alive. But this is the first step, and at some point when they divide and become these immortal kind of cells, they stop at the size of about 1 to 3 millimeters, and beyond that, they need to recruit the microenvironment. They need to recruit immune cells and blood vessels in order to get all the goodies they need, the oxygen and all the proteins and nutrients that they need in order to grow in size and have those blood vessels that will allow them to metastasize, to enter the blood vessel and circulate in the body and exit at a different site and cause a metastatic site. So I will share with you some of the data we have on melanoma and specifically on melanoma brain metastasis, but this is just one example of many projects in the lab that follow the same rationale, meaning that we look at the interactions between the tumor and the host, the tumor cells being those transformed cells, and the host, meaning us, the host for this persona non grata that nobody invited them to our lives. But if we understand these interactions, maybe we can sabotage them, meaning that we don't want them to occur, interfere with them, and then they will not progress. So if we look at melanoma as a study case, we have the primary tumor that at some point we have some melanoma cells that leave the primary site. The primary site usually can be resected by a surgeon, and then with follow-up, hopefully, if cells did not leave, we are healthy and we can continue living our lives normally. But if some cells did leave, they circulate and then they can exit at other sites. For example, what we found, and this struck me about 5 years ago, I heard a talk from a clinician from MD Anderson saying that they find in postmortem analysis of advanced melanoma patients, they found about 90% have occult, these dormant lesions in the brain. And I found this really disturbing because the brain is supposed to be a protected organ. I can understand why they go to the liver, why they go to the lung. These are the organs that get rid of foreign stuff or something that the body doesn't recognize. But for them to enter the brain, they need to cross the blood-brain barrier that usually takes care of the fact that these cancer cells or other foreign bodies will not enter. So our hypothesis was that these cells that leave the primary melanoma, we see the same, by the way, for breast cancer and for lung cancer, they all go to the brain. And we ask the same question, why? When they leave the primary site and they are now in the blood vessels, they secrete these proteins, these growth factors that can affect other cells. This is the way our body, our cells talk to each other. And when these factors, they can cross the blood-brain barrier, they cause the cells, these resident cells, normal resident cells in our brain to get activated, meaning that now they think there is something like a stroke or something that is out of the homeostasis that they need to react to it. And by reacting, they secrete other proteins, and those cause the blood vessels, that blood-brain barrier that was supposed to be intact, now it becomes leaky, and those melanoma cells or breast cancer cells or lung cancer cells, now they can extravasate, they can filter through those leaky vessels. It's like a tube with holes in it, and now they can live and then sit in this inflammation site that was created, and now they have all the goodies that after a week, a month, 20 years, they will start recruiting new blood vessels and immune cells and start growing in size. So the first step that we thought about was to deal with the primary tumor to prevent the whole process that I just described. And for that, we wanted to create an image-guided surgery probe. It's like a kind of a smart molecule that we will be able to inject prior to the surgery. And then once the surgeon comes to resect the tumor, there is a dilemma here, because if we have a lesion in the skin or in the breast, for example, the surgeon tries to not to damage too much of the healthy tissue, but at the same time not to leave residual disease. So he or she wants to be very precise and have exactly delineate the tumor boundaries and take as much as he can from the tumor without leaving any tumor cells, but at the same time without taking the whole breast out, just because we want to make sure that there are that there is no residual disease. So what we wanted to do is to create this probe that we injected. It is optically silent. That means that it is dark while it is in our blood vessels, but then when it finds a tumor, it shines out light, fluorescent light, and tells us, here, I found the tumor, and I'm releasing even the drug at the same time. So the surgeon in real time can see the tumor and cut around it. And what we saw is, at least in studies treating our tiny patients, those small mice, we see that we doubled the survival of those patients just by giving this probe prior to the surgery. So that the next step was to follow up on it, because at the end we know that there always might be some cells, soul cells, single cells that were left there, and we want to make sure that we have the right models to test on them new drugs that we develop. So we created some models of primary tumors that send metastases to the brain. Then we characterize them, we call it omics. It means that we do sequencing of the DNA, the RNA, and the proteins, it calls proteomics, and we see what is different between the metastases and the primary tumor, or the metastases and the normal brain, for example. And once we find, and I'm oversimplifying it here, but for the sake of the talk, if, for example, I found a protein X that is high in the metastasis, but it's very low or does not appear at all in the normal brain or in the primary tumor, this is a way to prevent metastasis. I will try to manipulate this protein, inhibit it, first by molecular biology, then by a pharmaceutical compound that I have or something that we develop as when we synthesize in the lab, and try to inhibit that protein and see if we manage to inhibit the proliferation, the migration of the tumor cells, and on animals to see if we inhibited tumor growth and metastasis. We try to envelope it, package it in a nanoparticle that will be like a missile that will take it directly only to the tumor and not to healthy organs. And then we test it on the tumor cells on the dish. This is called in vitro. Then in vivo in mice, what you see here is the brain metastasis in red because they shine light from the tumor cells, and hopefully next translate it to the patient, to clinical trials. But at that stage, around 5 years ago, we had some neurosurgeons joining our lab, and they said, why don't we reverse it all? Let's start from the operation suite, come to our surgery room, then we take a chunk of the tumor, the rest of course goes to pathology, and we do everything in reverse, meaning that we implant it in mice. Those mice are genetically engineered not to have an immune system that rejects the human tumor, because this is not a mouse tumor when we take it from a patient, and then do everything all the way in reverse. And this worked really nice. We all got promoted to professors, or the PhD students got their thesis, but we didn't really help the patients that we got the samples from. And this in a way was devastating because we worked so hard for several years, but each time it took about 6 to 9 months to have enough models, mouse models, to really test all the different combinations on enough cohorts to see that we are really able to inhibit the disease and prolong survival. Beside that, those mice, as I said, they are genetically engineered. They do not have immune cells or their immune system is defective. So we cannot test on them immunotherapy that are developed and is probably the latest revolution in cancer therapy, but we will discuss this later in my talk. So we had to test, to think out of the box, to think about something, a procedure that will be much faster, meaning that it will be a robust, rapid, and reproducible. And for that, we created a system where we get notified by the surgeons that there is a patient with a tumor coming to a surgery. We get the MRI prior to the surgery. We do the image analysis. Then we take, on the day of the surgery, we take the chunk of the tumor, we create cells, isolate cells from that tumor, and we put all the cells, meaning not only the tumor cells, but also those of the microenvironment of the same patient, and also the blood cells, where we have the immune cells from the same patient, and we 3D print them. We use a 3D printer to print like a mini tumor, or actually we print about 10 to 20 tumors, and then what you see here is that, we print them and we connect them to a peristaltic pump. We connect them to tubes that we can flow the blood cells from the same patient. So we have the immune cells and we can test immunotherapies. And at the same time, we can test different cocktails of drugs, existing drugs, or new combinations of drugs, or drugs that we invented in the lab. So this is how it looks. This is a 3D printed brain that we printed here according to coordinates that we got and converted from an MRI. But more importantly is what you don't see inside here. So this is a gel that really mimics the tissue properties, the mechanical properties of a tissue, meaning the stiffness, the swelling, all the properties that are different between a tumor that comes from the breast, from the brain, from the lungs, from each one. And we have a gel that mimics each one of those organs. When we print it, the cells live in this gel for around 2 months at least. And we make sure that we have functional blood vessels that we print with the blood cells, with the endothelial cells that are from that patient, from the same tumor. So what you see here on the left is blood vessels that we print that are similar to the blood vessels in contact and in structure of the blood vessels that was an hour ago in the patient's brain. And then you can see that they are functional. So we can flow. What you see in green is that we flow the blood. In this case, we flow the blood with a dye, a polymer that is bound to a fluorescent dye, so to make sure that you can see how it flows inside. And here you can see the different cells. So we have a full brain as opposed to a full tumor. In this case, it's glioblastoma or brain metastatic melanoma. You can see in green are the blood vessels, the endothelial cells. In blue, you can see here the resident cells in the brain, and we have the cancer cells in red. And here you can see how we take the blood, only the blood vessel out of all these tissue that we see on the top right, and you can see where we can flow the blood from that patient and test different drugs on it. So this is a much more reliable system that we can test our hypothesis than on just tumor cells on a Petri plastic rigid dish that has nothing to do with what we see in the body. And then we are usually surprised that things worked wonderful in the lab, but they did not translate to the patient when we did clinical trials. And this is partially the reason. We don't have good enough models, and we hope that these models will recreate better the clinical setting. So not only do there are functional vessels, but you can see here that we mimic the vessels that have inside the tumor cells in the brain that go out and then they sit in the brain as a metastasis. This is how we do the translation to the 3D printing. So we take all these models, and when we have these models, we can study the interactions between the tumor cells and the microenvironment, the immune cells, and the rest of the blood vessels and the rest of the brain stromal cells. And according to what we find, those markers that we find, during our trying to take our hypothesis and find the suspects that we would like to inhibit, we create vaccines to prevent the metastatic process, or in high-risk populations, we can maybe prevent the occurrence of the disease, like in BRCA, the breast cancer gene, that today all we can offer is to double mastectomy, both breasts. And later, most patients come with already a disease. We would like to interfere and even regress if it's already a systemic disease. Now, to combine what we find with the immunotherapy, we wanted to look up to when we see here in 2018, the Nobel Prize in Medicine and Physiology was awarded Jim Allison and Tasuku Honjo, that were the pioneers in finding those immune checkpoints, meaning that those molecules that you see here, two of them that are relevant are the PD-1 and CTLA-4, that there were antibodies that were created against them to inhibit them, and in a way, they inhibit the interactions the bad interactions, meaning the immunosuppressive interactions, when the immune system is not doing its job. The T cells, the immune cells, are not attacking the tumor, but rather when they connect with the tumor cells, they are inhibited. And this is a real revolution, but this has limitations as well. We see that there is a low response rate. As remarkable as it is, we still talk about 20 to 30% of patients that respond to immunotherapy, and it only in several cancer types like melanoma and lung cancer and renal cell carcinoma, but there are others like brain cancers, like the glioblastoma that we work on, or pancreatic cancer that do not respond. And we wanted to deal with that, to improve and make them more sensitive to immunotherapy. At the same time, we see that they develop resistance and that there are some severe side effects because we are overreacting the immune system. So our idea was to create a nanomedicine, a nanovaccine that in combination with the immunotherapy, those antibodies, we will prolong the survival of the patients and improve it. And in a manner that has decreased toxicity, we can give much less in terms of the dose that we need, and this will be a very specific effect against the cancer. So this is a very complicated slide, but to make it really easy, what we did is we created those nano vaccines. These are nanoparticles. They are around 200 nanometers in size. We give them to the patient. Again, our patients are the little mice at this point. And once we inject them, the immune cells, those purple cells, they swallow those nanovaccines. And the nanoparticles, those nanovaccines have these little proteins, structures of proteins that exist in the tumors that we have identified originally in our scheme that I presented in the beginning. So once they are swallowed by the dendritic cells, those cells of the immune system, they are activated, they go to the lymph nodes and they activate T cells, that these are kind of the soldiers that go out from the lymph node, and then they produce, on one hand, we have a cell response. It means that the T cells go to the tumor and kill the tumor cells. And also we have the B cells and the formation of antibodies that will recognize the tumor cells or markers on the tumor cells. And remember this point when I'll switch to COVID-19. So we took the immune cells from mice that we vaccinated them and put them together with the red cancer cells. And what you see here is that the immune cells that came from the vaccinated mice, they come and they eat, they kill the tumor cells. So we were confident to continue to animal experiments at this point. And what we did is we injected 3 times our vaccines together with immunotherapy. And sure enough, lo and behold, we saw that we inhibited tumor growth and we prolonged significantly the survival of those tumor-bearing mice compared to what the immunotherapy alone can do. So basically what I showed you here is our approach, a multidisciplinary approach that we start from the patient bed, we go to the lab, the biologists, the MDs, the chemists, the bioinformaticians, all together work together to get it back to the biologists to test it, and then from the bench go back to clinical trials. So how does this connect to the current pandemic, to COVID-19? So I'm sure all of you are familiar with this structure already. This is the SARS-CoV-2 virus. And what is important here is to look at those spikes. This is the spike protein. There are other 3 proteins called the nucleocapsid that is inside to the RNA. This is the longest RNA virus that we know. And also the envelope and the membrane proteins. And those 4 proteins are called structural proteins. And against them, mainly the spike, are all the vaccines that are being developed currently, like the Moderna or the CanSino and the BioNTech and many other companies are developing against this spike protein so that there will be antibodies formed in the body against the spike, those proteins that stick out, because once the virus will infect or try to infect the cells, there will be already antibodies that will bind to these spike proteins. So this is the mechanism of infection. I'm sure even in this webinar series, you already described it. And what I wanted to show you here is the way that the virus binds to its receptor on the lung cells in the alveoli, for example, also in other places, but we can discuss this later. And there are a lot of different drugs that are trying to be repurposed to inhibit different states of the mechanism of infection until the creation of new virions that you see here, that then at the end of the process are being secreted from the cell and going to infect other cells. So what we did is to create the same vaccines that I just mentioned for melanoma and other cancer types, only that this time we put inside, instead of these tiny pieces of proteins that are associated with cancer cells, this time we took tiny pieces of those proteins like the spike protein. And the idea is exactly the same process until we get antibodies. So then once that the body will see a virus, it will attack it and bind to this binding domain here on the spike and not allow the virus to infect the lung cells. So where are we at the moment? We already vaccinated about 500 mice. We saw that we managed to create antibodies. Those are specialized antibodies that are very specific at high affinity to the virus, to certain proteins in the virus. And we are now testing them to see if they can neutralize the virus. And the problem is that there is no real model of disease. And we can discuss this later, how fast we are immediately translating now to patients or to healthy volunteers in clinical trials. I think this is probably the fastest ever that we saw. Ebola and measles were developed in probably record time in around 4.5, 5 years. So now we are trying to do it in less than a year. This is quite a challenge. So if I go back to cancer, what we usually do in our lab, back to the same platforms of vaccines, is if 100 years ago, we would tell someone that above the infectious place, site, 10 centimeters above it, amputate the arm, for example, and then Fleming came in 1920s and identified the penicillin. And since then, we have a lot of antibiotics developed that we can give even orally or intravenously without even knowing where the infection is. We hope that within 100 years, we will be able now to get to a stage that we can create these nanomedicines that will be used for early detection and also for prevention and cancer therapy to transform cancer to a disease, basically cancer without disease. So this is my fantastic group. I love each one of them. They're really bright, coming from biology, medicine, engineering, and chemistry. The way we look and attack cancer or COVID or SARS-CoV-2 is in a multidisciplinary way, because we really believe that when you drain the water from the ocean, you'll find out that all the islands are connected to each other. Thank you. Jennifer, ready.
Thank you, Professor Satchi-Fainaro. Let's start a little bit with some questions about your work specifically, and then move to some questions we have about the coronavirus. You made an exciting announcement last year that your team had developed a nano vaccine for melanoma. Can you tell us a little bit more about this, including what's the difference between a nano versus a regular vaccine?
Okay, maybe vaccine is not the right word, it's just easier to understand. First, a nano means just the size. Nano is 10 to the minus 9 of a meter. It's very small, but it's small for us. It's tiny. The vaccines, the molecules that we designed are around 200 nanometers in size, so they are really tiny. If we talk about nano, 1 nanometer to a tennis ball is like a tennis ball to the earth. Okay, this is just to get the ratio of the size. So it's only for us, but if we look from the body size, this is huge for a blood vessel. So from a normal blood vessel, it will not leak out. It will leak out only at sites of inflammation. It will leak out from blood vessels only at sites.
I'm sorry, we seem to have some technical difficulties from Israel. We are trying to reconnect. Stay with us for a moment.
Can you hear me?
Yes, we have you back.
Disconnected. Okay, sorry. So what I was saying is that if we compare a nano vaccine to a regular vaccine, it's a matter of the size. First, it's a nano, it means that it's 200 nanometers in size, but much more interestingly is usually the way we look at vaccines are against infectious diseases, mainly viral diseases, and we take an inactivated or a dead virus, and we activate the immune system by injecting that. In our case, it's completely synthetic. So we can control the design and know exactly what we have in it and target it specifically to the cells of interest. So we don't get a general effect in the whole body, but only on those cells that will at the end produce the antibodies against SARS-CoV-2 virus or against the specific tumor cells and not something that is general.
Wonderful. Can you tell us where are you in the process of developing this treatment, and any idea when you think a person would have access to the treatment?
Okay, this takes a really long time. Usually, it can take up to 5 or even 10 years. The process of drug development in general, as I mentioned, for the record time that we had for the Ebola vaccine, for example, this was 5 years. And now we are working way faster. I think also the regulatory agencies are very supportive, trying to make everything much faster with some risks as well. But I hope that we are in the stage already that we can manufacture it in grams in the lab. Now we need a company, and we are creating a spin-off company that they will be able to manufacture it at kilograms scale. So we need scale up and to do it in GMP, meaning that it will be sterile and in a way that can be performed for clinical trials. In the university, we started the innovation part. The rest we give to a company to continue that and progress to patients. Hello?
We can hear you.
Okay. Yes? Can you hear me?
Yeah, we can hear you.
Okay, so I said that we do the innovation part in the academia, and then we give it to a company via Ramot, the tech transfer office that takes it further now to a company that will scale it up and create it at a sterile form that can be injected to patients and deal with the clinical trial.
Okay, let's talk a little bit about, you mentioned in your presentation that we all have dormant cancer cells, or some percentage of people have dormant cancer cells in their body. Can you just explain a little bit about, does everybody have them, or do only some percentage of the population have them, and what activates them? We have a question from one of our listeners.
Yeah, that's the million-dollar question. So I think in general, some people have them, some don't. We always, if you think about it as a factory, our body as a factory, it's like a factory that we always have the rule of 1,000, is that if you're producing washing machines, always one of in every once in a while will go wrong. Usually I buy that, I don't know, it's the luck I have. But then it means that there should be there is a QC department that will make sure that this doesn't leave the plant, right? But the same goes in our body. There should be this QC, these regulators that check that everything is going according to plan and is normal. But sometimes they miss. And when they miss, we get these different mutations that, as I said, in some percent of the population, in some cases, in some organs is higher than in other organs, but we can live with those because our body knows how to deal with them. You saw 99% in thyroid carcinomas, they are actually not awakened and continues to become a clinical problem. We live with with not even knowing that it's there. And this is how we want to keep it. So I think in a way, what we have to look is, if you're asking the reasons and what awakes them, there was a fascinating paper about a year ago from Vogelstein, 2 years ago already, from Vogelstein, that got the Dan David Prize in Tel Aviv University, that said that there are a lot of different reasons, like hereditary and UV radiation and carcinogens, but 60%, 65% is just bad luck. It's many things that happen at the same time and we cannot control them. There are some that we can control. There are some that we can do follow-up if we are in high predisposition for cancer, but many others is just a combination of many factors. And we can control some, but not all of them. And I think follow-up, screening, this is the way to deal with it without being terrified on a daily basis. We have to live with it. And usually, as I said, our body knows how to deal with it and not to let those dormant lesions wake up.
Thank you. We have a question from our board member and member of Board of Governors, Gary Rand, asking about whether you're looking at specific cultures of differentiation to activate or deactivate as a strategy.
Yes, I'm not sure I understand what is specific cultures, but if it means from the immune cells or the cancer cells, so we work on specific populations of the immune cells. We isolate them, and then we run what is called single cell RNA sequencing. So we go and really identify specific cell populations. So we look at specific tumor cells, and we know that every tumor is really heterogeneous. So even if you take different cancer cells from the same tumor tissue, they will not be similar necessarily. And I think in a way, this is one of the reasons that it's so difficult to cure cancer, because it mutates, it changes all the time. We try to isolate one population. For example, if we look at breast cancer and we did the histology, the pathologist told us, okay, this is a HER2 positive cancer. It has that marker. We will give a certain drug, an antibody that kills all the cells that have this HER2, but there will be some other cells in the same tissue that will not have that marker, or that with time it changed or mutated and have other markers that gave them the advantage of surviving. So we are all the time, the cancer is tricking us and we are tricking it back. So it's like a game that we try to evolve together with the special populations. I'm not sure if this is what what he meant, but I hope I answered the question.
The question was about clusters of differentiation?
Cluster differentiation, yes.
Clusters,
So we
yeah.
do that, absolutely.
Yeah. Wonderful. Okay, let's move to, now we're going to bridge a little bit to the COVID-19. What are the, you've been doing work and really having breakthroughs with regards to melanoma and other cancers. What are the similarities between the cancers you've been studying and the coronavirus?
Okay, actually, this is the work that we've been doing in the last 4 months, which is nothing in comparison to 20 years of work on cancer, but the similarities are striking. What we found is that usually we thought that against viruses, we need antibodies, antibodies, antibodies. We never took into consideration, at least when we designed it, that the cell response will be so important. And what we see here, what we saw from our studies in cancer, is that first we targeted in cancer, we targeted the cell response. That means that the immune cells that we activated with our vaccine, now they go to the cancer cells where they identify the cells that have this protein that I said that we put in our vaccine, and they identify it and kill it. They kill it by secreting several, let's call them toxins, proteins that and other analytes that kill the cancer cells. So this is one mechanism. It is called the cellular immunity. And this is immune cells that secrete factors that kill the tumor cells. Then there is the other arm that is called humoral immunity, that this is the B cells or those that create the antibodies. And the antibodies is what we always looked if we manage to create antibodies. And there are different types of antibodies. And what we saw that in the cancer studies that caused us to think that we can even deal with SARS-CoV-2 is that we saw that it had memory, meaning that those mice that we vaccinated, once we challenged them again with cancer, the cancer didn't develop. It's like the immune system remembered those tumor cells, and now when they came back, it prevented metastasis, it prevented the transformation of new cancer. So that caused us to think that it will happen the same with the virus. So we did the same, but what we found during this process is not only that we created both arms of immunity, the cellular and the antibody one, is that amongst the antibodies, we created a high level of special antibodies that are coming from the germinal centers in the lymph node. And these are antibodies that have a high affinity. So we don't need a lot of them, but they are very strong, very potent. And we saw that we managed to get them to be established. And this seems to be really important especially for vulnerable populations that are more prone to become critically ill or be in the severely ill patients for COVID-19. So they are not able to create many and strong immune response, but if with the vaccine we will manage to create small amounts of antibodies that are very potent, that will hopefully be a nice surprise.
A few weeks ago, we had a webinar with Professor Gershoni, and he told us about vaccine development and basically said it's very difficult to rush vaccine development. There's all kinds of stages of clinical trials and human trials. What do you think a sort of realistic timeline might be for a COVID-19 vaccine? Any help you could shed on this matter? I know it's something everybody is wondering.
Okay, for the Hebrew speakers, הנבואה ניתנה לשוטים. This is we are all experts of the past. It's very difficult to predict the future, right? So to think how long it will take. As I said, we have experience with Ebola and measles that was the fastest, as I mentioned, but I think here we're dealing with much faster developments, but bearing in mind that most of the vaccines that we are talking about, these are vaccines that were a type of vaccines that have in them either DNA, RNA, and peptides together with different, they are mainly based on nanoparticles. and not on the weakened or dead virus in this case. So these are synthetic and they do not exist yet, meaning that the platform exists. It existed for other diseases such as cancer, and it delivers the RNA of that protein, for example, of the spike protein. This is the Moderna vaccine, but it's not approved yet even for any other indication. So on one hand, we have the platform is really promising, and hopefully we're talking about a timeline that will be between a year to 2 years. But I think that the best vaccine is yet to come, because we don't have enough time to test different combinations of, for example, of the peptides or of the RNA of different proteins, not only the spike, and so on. There are so many things, different adjuvants. So maybe with the first vaccine, it will be ready in a year, but probably there will be other vaccines that will be developed for SARS-CoV-2 that will be more potent later on.
So we could have a situation where there's like a sort of a rolling cycle of vaccines, new vaccines coming to market that get closer to meeting the mark of actually really dealing with the COVID-19 outbreak.
Right, and I think this diversity of vaccines, of vaccine compounds, each one is different, is really important. And science is so advanced now technologically that allows us to synthesize such vaccines that, for example, one of the things that really worries the WHO is tuberculosis. We have only one company in Denmark that produces one vaccine for tuberculosis. And once there is a shortage, there are no vaccines for tuberculosis, and we see an increase in the disease. So I think it's really important to encourage many companies, many academic labs to come with new ideas and to develop new vaccines, because there will be a need. It's not that there will be one vaccine that will be out there and that's it. We all wasted our time. And I'm very optimistic about it.
Have we seen the nano vaccines being used for any diseases in human trials yet?
Not yet, not ours. Ours is just now being translated to a spin-off company that, as I said, they need to scale it up and create enough of it and start clinical trials. But this alone will take about a year.
That's another question we had from one of our listeners about partnerships. Your lab having partnerships, I guess, to help bring the drugs more quickly to trial. Are you working with?
Yes. So for example, on the COVID-19 vaccine, my lab won a grant from the Israel Innovation Authority that requires a partner. So Merck partnered with us to see the results. They are allowed to see the results in a year time. So we advance enough for something that hopefully they will take it further.
Thank you. Let's end with a few specific questions relating to the coronavirus. First of all, question about if someone has had immunotherapy for cancer treatment, do they have a better chance of being immune or protected from COVID-19?
This is a fascinating question, actually. Usually the answer would be no, there is no relation, no correlation, nothing to do with it. But as we're just publishing, actually in 2 days, we have a review on immunotherapy for COVID-19 that will be published in Nature Nanotechnology in 2 days. And what we saw there, we looked, while designing our nanovaccine, to do it in a rational manner, we read a lot of manuscripts. There was a bombardation of manuscripts. 25,000 papers were published in the last 4 months on COVID-19. This is unprecedented. So we had a lot of reading, and as I said, we have 30 people in the lab, 15 of them were at home. So all were reading a lot and summarizing and creating all these tables that in some of the studies we saw, and we summarized it in this review, I'll be happy to share with you as soon as it is published, is that some of the immunotherapies, like the anti-PD-1 that I mentioned that won the Nobel Prize 2 years ago, is actually being tested in clinical trials for COVID-19. There is a rationale that it will protect because of some of the way it deals with the immune cells and how they deal with the virus or any foreign body. So the same rationale that I mentioned also worked here in a different manner, but might be helpful. So anti-PD-1 or anti-PD-L1 are in clinical trials. There are other antibodies also that are tested as immunotherapy for cancer, and now they are also or treated already for cancer. We see it in clinical trials for COVID-19. So this is really interesting, and it might be that this patient is actually protected in some cases.
Speaking of antibodies, we hear about antibodies. There are multiple different types of antibodies. Some may be longer lasting, some may not last. Can you just give us sort of the roadmap as to antibodies, and are they going to protect us against the disease? Will they make us immune? Will they last?
Here there is no simple answer. Usually we would say, yes, if there are antibodies that were formed after you were sick with, you were infected with the virus, these antibodies should be protecting you from recurrent infection. However, we do know that, for example, with the influenza, we are being immunized every year. It's not that we have one shot and that's it, we are protected for life. For many reasons, some are mutations and some are seasonal, that every season we get a different combination of SARS, and we don't know yet what will happen with the SARS-CoV-2 in the next cycle, in the next winter, for example. So far, it seems that there are mutations, but very little and in certain areas. So out of 1,076 patients in the US that were sequenced for the SARS-CoV-2, only 4 had mutations, and these were very small mutations, meaning a replacement of one amino acid by another. This is not something that a vaccine doesn't know how to deal with, because it's not in the area that the vaccines are usually developed for. But having said that, there is another study that showed that some of them are longer lasting, of those antibodies, and some are short-lived. And amongst them, some can be only 21 days, as far as 21 days. So it seems like they are there, but they are not potent enough to protect, or are they disappearing and we need to give a boost? So nobody knows there yet if there is really a secondary infection, if this really occurred. In some cases, it could be that the people were still sick, meaning that the test that they were given as a negative result, or the serologic study showed already that they have antibodies, was either false positive or false negative in correlation. So I think it's early days, but again, we are, all the scientific community is working in such a collaborative manner. And we're talking about scientists in academia, in the industry, in the clinic, everywhere putting their hands together. I just hope that this way that everybody works together on sharing information, sharing data, that it will only continue this way after the pandemic is behind us.
It sounds like that's such a wonderful note to end on. It's great to see that there's been real collaboration across both the university and across the country, across the scientific community worldwide, to really try to deal with this pandemic, which has been the first we've experienced in our lifetime, and certainly in the US, and has caused so much pain for so many people. Thank you for being so generous with your time, Professor Satchi-Fainaro, and for sharing your insight into your work with us and the applications for COVID-19. We thank all of our friends from across the country and from around the world for taking the time to join us today for this webinar. We will send everyone a link to today's presentation so you can learn more. I know a number of our visitors were interested in getting the presentation, as well as links to learn more about the work being done in Professor Satchi-Fainaro's lab and the fight against coronavirus at Tel Aviv University. Please stay safe and healthy, and we look forward to seeing you at our next webinar.
Thank you for hosting me. Keep safe.